very grateful for the organizers organizing this conference and for asking me to speak. And also for several expressions of personal expressions from some of you at a difficult time for me. I'm going to tell you a story today about theorists' view of protein phase separation based on in vitro experiments. So the story is very related to the experiments and motivated by it, very appropriate for the theme of this conference. But it's a story, and the story has several anchors that are based on experimental data, but the story also has story. And at the end of the story, I will put up some questions that are forward-looking that maybe you want to think about and especially measure to see how much of this story is general, how much of the story is accurate, and how much of the story is important for biological processes that people are interested in. The experiments are done in the group of Daniela Goldfarb at the Weizmann Institute on one protein, CPEB4, and in the group of Tony Hyman and Joy Carr when he was a postdoc in that group on FUS. The papers are published over here, and I won't keep showing you them. And what I'm going to talk about today, which is the story, is in this paper that was just published a couple weeks ago, but is based on the experiments in all of these. I also want to thank the Volkswagen Foundation for their support of this research. Okay, so changing the paradigm, proteins are not necessarily distributed in a dilute manner as what we might call an ideal gas or ideal solution in the cell, but can form condensates, and everybody here knows this. And just to advertise other work that we're doing on condensates in the nucleus having to do with chromatin, this is not a picture. These are actual confocal microscope pictures of chromatin phase separation in Drosophila in live flies, and the flies are actually alive. These are muscle cells, and we have worked with them on phase separation of chromatin, and as you know, chromatin is DNA that is linked together with proteins. So this happens if you just put DNA in the nucleoplasm. Nucleoplasm is a good solvent for DNA, and this happens because of the proteins. And I'm not going to go into the details, but I'm happy to discuss this privately. This is very recent experiments with the group of Gary Karp in Berkeley, with whom we're collaborating, very in vitro. This is very live flies. This is very in vitro on short oligomers that represent heterochromatin, where the K9 site is on the histone tail is methylated, which is the red, and the blue is unmodified chromatin oligomer that shows phase separation and buffer. So this is just advertisement, but I'm happy to discuss this. What I'm going to talk today about are phase separation of proteins in vitro experiments, where even in the phase, which is supposedly homogeneous and not phase separated, clusters of proteins are observed that are very big. These clusters are much smaller than the micron-sized condensates, but they, in this case, a FUS can still contain thousands of protein molecules. And the question is, why do these occur? Are they just a fluctuation? And in FUS, we believe they're a type of fluctuation. But in the other protein, CPEB4, we think they're a rather stable state, and I'll try to show you why we think so based on experiments. Okay, so, okay, here, condensates, pictures from, is Alexandra here? Oh, okay. I'm going to talk about some of her data later on. The proteins are very dilute. They form condensates. This is all in vitro because of some kind of attractive interaction between the proteins. And then if you see these clusters with hundreds to thousands of proteins that are observed in the single phase regime where the system is thought to be macroscopically homogeneous, why do they what do they happen? If you just mixed water and alcohol and you play with the temperature so they phase separate, if you look in the one-phase regime where there's no phase separation, yes, you'll see alcohol and water molecules. You may see dimers of alcohol, trimers, but you're not going to see fluctuations with hundreds of molecules unless you're very close and very, very close to a critical point, and these experiments are messy enough that they're not close to the critical points as far as we know. And the story here and the message is that we think these clusters are stabilized in a system of protein, water, and salt, very simple system, by proteins that can have several coexisting conformations. So we'd all say, okay, of protein and water and salt, it has some conformation. These are intrinsically disordered proteins, so they're polymer-like. And what I'm going to try to argue and show you also based on experiment is that in the same system, you may have proteins that are mostly hydrophilically conformed. Their conformation allows them to be soluble in water. Hydrophobically conformed, where they want to escape from the water. And amphiphilically conformed, where the protein has a blob in the sort of sense of polymer physics, which is hydrophobic, and another blob that's hydrophilic, and I'll show you the experiments. And we're going to also have two paradigms for LLPS, and whether these protein clusters aggregate as clusters and remain in clusters in the very large LLPS domains when phase separation occurs, or if these proteins just coagulate in the way that people would normally think happens. And we'll see the implications for low surface tensions. So let's go ahead. All right, so protein physics. Proteins are linear macromolecules, chains of many small amino acids. I think everybody here knows about amino acids, and some are hydrophobic and some are hydrophilic, and many proteins are folded, so their nature is rather fixed and their chemical nature is known. However, there are disordered proteins that are polymer-like and are fluctuating. And can these proteins behave as polymers, but not as homogeneous polymers where every monomer is the same, but with polymers with regions that are fairly large, larger than few monomers, that have some chemical identity, that are more hydrophobic and less hydrophobic, and we think that is the case. All right, so this is just a schematic of a phase diagram where the x-axis is a fraction of solute and the y-axis is temperature, and you know that you have two phases that can coexist if you are in equilibrium. And this phase diagram defines the saturation concentration at any fixed temperature, namely in this region, in what we'd call the one-phase region, you are below saturation and your system is not phase separated. And again, we're going to see that this very simple picture, where there's no reason here based on statistical mechanics of simple solutions to expect very large clusters, might be different in proteins that can have several types of conformations at the same time and in the same system. That can give rise to mesoscale structures, even in the subsaturated one-phase regime. And here are experiments on the protein CPEB EB4. It's the protein minus the RNA binding domain, so it's just the disordered part of the protein in water and salt. And again, the subsaturated region we're going to focus on in most of the talk is here, where you don't have phase separation. And these are dynamic light scattering experiments. The radius of gyration of the protein is a few nanometers, but yet these clusters have a typical diameter of, 30 to 100 nanometers, depending on the temperature. At about 20 degrees, I think they phase separate. So this is all in the single phase regime where you get these very large clusters. And AFM imaging also shows you these clusters, if you like to see real space pictures. And those of us that are old were brought up in Fourier space, so we like dynamical light scattering. All right, the phosphoprotein, that was CPEB before, and I'm concentrating on the two because they're the two proteins our collaborators studied, but we'll see that we think they're two different paradigms. And the first protein also shows clusters. Those clusters are significantly bigger, as you can see here. They're hundreds of nanometers, 250 nanometers. Again, the protein radius of gyration is a few nanometers. So FUS shows much bigger clusters. CPEG shows smaller clusters. And they're both just protein, water, and salt. Now, large clusters appear in subsaturated regimes, and when can we think in sort of simple physical systems of mixtures when we might have large clusters that are stable in a system which is not yet phase separated, it's subsaturated? That's something called microemulsions, and I don't think too many people, maybe besides Shura, know me from my previous life at the Exxon Corporation, but many of us worked on microemulsions because they were important to the Exxon Corporation. Well, we worked on them because they were fascinating as far as their physical chemical properties are concerned. And these are droplets that can be water in oil with a monolayer of a surfactant or an amphiphile at the interface. An amphiphile has one part of the molecule that's hydrophobic and one part that's hydrophilic. They could also be oil and water systems. And questions are, we see something similar, somewhat stable droplets. Maybe emulsions, maybe they're metastable, but they seem to be somewhat stable. But we don't have any amphiphile. We have no soap, and we have no oil. So how can we see that? And we think we can see that because the very same protein can play the role of a hydrophilic protein. The oil, a hydrophobic conformation. When I say hydrophobic, it's not oil. It's really less hydrophilic and can grab some water along with it. And amphiphilic, which is a combination of both the more hydrophilic and the less hydrophilic. And there have been some very interesting recent papers on coexisting conformations of disordered proteins, looking more molecularly at the protein conformation. So this is not a completely crazy idea, but I think we had the crazy idea before these papers, and we were very happy to see them. But we're going to bypass the conformations, because that's not our expertise, and that's not exactly what the experiments were studying. And we're going to see, can mesoscale experiments, light scattering, dynamical nanoparticle tracking experiments, which is looking at the sizes, can they tell us something about these clusters, even though we don't know in detail what these conformations are, except for the story I told you? Okay, so I'm not going to show you this. All right, now, a very, very important clue in the story, and this is a real experimental anchor. Our EPR experiments by my colleague in our department, Daniela Goldfarb, who's an expert on magnetic resonance and EPR, and she's done solids, liquid crystals, and other systems. And what she observed in the CPEB4 protein was 3 different dynamical behaviors of the same protein in solution in the single phase regime, where you have no LLPS. So she has this protein of water and salt. EPR, they attach a spin label to part of the protein, and that protein is doing rotational diffusion. And the time that they can measure, the relaxation time, is related to the rotational diffusion time of the protein. And as you can see here, at various temperatures, she measures 3 different rotational diffusion times that differ by a lot. This is a log scale, so they differ by about 2 orders of magnitude. So some proteins are rotating quickly, some proteins are rotating very slowly, and some proteins are rotating with some intermediate time scale. Significantly, and this will be important for the end, so just remember it for the end, here's LLPS in the system, and these time scales change around LLPS, but there's no signature of a phase transition as far as these time scales of rotational diffusion constants are concerned. They change very smoothly through LLPS. So this led us to our story and a picture that we can try to justify, and we have justified on the mesoscale, but not on the molecular scale. And so here's our disordered protein. and it has hydrophobic regions, hydrophilic regions. If you will want to see more about the particular proteins and how many hydrophobic and hydrophilic amino acids, you can ask me during the questions, but that's not our expertise. And we said, well, you can have red as hydrophilic, blue is hydrophobic. You can have this kind of hydrophilic conformation where this hydrophobic is probably somewhat collapsed, but we didn't draw it as completely collapsed because it's not really, it's less hydrophilic, it's not oil. So it's more collapsed than the red one. We can have the opposite where the hydrophobic's on the outside and what we'd say the more hydrophilic is somewhat collapsed on the inside, probably brings water along with it. And then we can have an amphiphilic one where you have a blob that's hydrophilic and a blob that is hydrophobic. So this is an assumption on our part, but the assumption is based on those three different time scales. And now these clusters are imagined to, the system is imagined to have clusters. Here's a cluster, and you have the hydrophilic conformation with the hydrophilic part, the red one, on the outside, and that has fast diffusion. I think that's intuitively obvious. You have the hydrophobic or less hydrophilic conformation on the inside, so the blue is on the outside and the red, the more hydrophilic on the inside, forming the core of this cluster. And that's maybe in sort of a melt or in sort of a, because it's probably water, probably in some kind of fluid, and it's less, has less rotational freedom than these. And that we identify with the intermediate time scale that's been measured. At the interface, you have, we suppose, you have this amphiphilic conformation, and that we associate with the slow time scale. Why? Because these are rotational diffusion constants. You're oriented here by this blob wanting to be in the water, and this blob wanting to be in the core, and you can't just rotate because then you would violate the hydrophobic interactions that are causing this orientation, like a lipid at an interface or a soap molecule in interface in the system. So these are the slowest rotational diffusion time, and that's known also for liquid crystals and other systems. So based on the three measured times, we propose that these clusters have this structure to them. They're not just some blobbish thing, but they have a substructure to them. And there's a black copolymer analogy for everything I've said, but proteins are more complicated than black copolymers, but we're still abstracting them to have a mostly hydrophilic, less hydrophilic, and then to have two blobs with the amphiphilic conformation. And Gonain, my collaborator, I forgot to mention him, Gonain Golani was a postdoc in my group, and he is today a faculty member in the University of Haifa. We're trying to see what analogies we can make between black copolymers and these proteins, but they're just sort of analogies. All right, so now, people did light scattering, people did nanoparticle tracking analysis, and they also directly observed through AFM and electron microscopy these clusters in the single phase regime again. And we wanted to see, can we say something about the cluster size distribution? So we are taking the chicken's way out. We're bypassing the conformational problem. We know so little about these, and so little is known experimentally about these supposed conformations that we're not trying to predict them from some first principles, or at least today, even from a black copolymer model, although I hope that'll be done later. But we're saying, what can we say about these clusters just on the basis of their geometry? Well, they have an energy associated with their core, with their volume, R is the radius of the cluster, an energy associated with their interface, the interfacial tension, and an energy associated with their surface that has to do with their curvature. If you have these amphiphilic blobs at the interface, they may be packed with the right curvature, or they may be packed with the wrong curvature. Those of you who come from membranes know that's known as a spontaneous curvature of the interface, if the interface likes to point towards the water or towards the oil. And when you go through the mathematics, this is R cubed volume, this is surface R squared, this is R, and there's another term that's a constant as far as the energy per cluster, which is just the bending energy, not whether you want to point towards the water or oil, just the fact that you've taken a layer, an amphiphilic layer, and you've bent it. Doesn't matter if you bend it towards the water or towards the oil. There's also another term, the energy, but this K, kappa, doesn't depend on cluster size. So it doesn't enter into the cluster size distribution that you would measure in light scattering. And the probability of having a cluster of size R is related to the Boltzmann factor of this energy. It's an equilibrium system from everything that we know, or at least long-lived medicine. Now, there's an idea in self-assembly of such clusters called the critical aggregation concentration, which is if you have very, very few molecules in the system, then the molecules have highest entropy when they're just dilutely distributed in the system. But if they tend to aggregate because of these hydrophobic interactions, there's a certain concentration at which the entropy, the translational entropy of just having individual molecules in solution is not, the gain in that entropy gets less and less as you increase concentration, but then you gain from the energetics of trying to hide the hydrophobic parts from the water. And that concentration is called the critical aggregation concentration. Those of you who've heard of micelles, there's a critical micelle concentration. It's very, very analogous, but for more complex aggregates like the one I described. The theory tells you, the theory of self-assembly, below the critical aggregation concentration, when you're very, very dilute, you have few clusters. You do have them because they are stable energetically, more stable energetically, and there's some probability to have them. And if you go through the theory, the size of these clusters will increase with concentration. Above the critical aggregation concentration, you have some cluster that's fairly stable energetically. Most of the proteins in the clusters, not in monomers in solution, and the radius stays almost independent of the concentration. This is very standard statistical thermodynamics of generalizations of micelles. Okay, this is like a generalization of the micelle with the surface and the core. The experiments tell us that in FUS seems to be below the CAC. The experiments indicate that only 1% of the protein is in these clusters. So there's a beautiful paper in PNAS by Tony Hyman and Joy, but you have to read that paper and say, oh, we discovered clusters in the subsaturated regime. That's great, but it's only 1% of the protein. The CPAB-4 protein seems to be above its critical aggregation concentration. Most of the proteins are forming these clusters, and this 90% is gotten by quantitative analysis of these rotational diffusion times and the amplitudes in the EPR signal as to how much protein is fast diffusing. Those are the monomers in solution, and the ones that are slow rotational diffusion in the clusters. So we see from the data that we have three diffusion times. We say that gives us 3 coexisting conformations, and people are looking at that more on a molecular scale. And then we have this self-assembly idea. We try to analyze the cluster distributions very phenomenologically. We don't know the constants that precede these, but we determine them from the experiments. And then from some very qualitative measurements, quantitative measurements, but on qualitative grounds, we see that FUS, only 1% of the proteins in clusters. That means these clusters are basically fluctuations, but they're fluctuations that have some energetic stabilization. So they're not fluctuations that have a probability of 0.0001%. In CPAB-4, these are sort of stable objects, because most of the proteins are there. You tell me when I have 5 minutes left. Now? Okay. So another thing that I noted is that below the critical aggregation concentration, when clusters are just fluctuations, the radius of the cluster will increase with concentration, and that indeed is the case for FUS, and there's a scaling law you can predict that, within the error of all the experiments, maybe is obeyed. In the CPEB, in the other paradigm, where you have 90% of the protein in clusters, so these clusters are more stable objects, the radius very weakly depends on concentration. So we see these two paradigms, clusters as fluctuations, and very few of the proteins are there, or clusters as more stable objects with most of the proteins there. We did fits to the light scattering data and also to nanoparticle tracking analysis, and I just don't have the time to tell you in detail about the fits, but I'm happy to talk privately. In the paper in Communications Physics, published very recently, all these fits are there with the discussion. You might just want to note that in the CPEB EV4, this is the probability over the probability of the peak of the distribution on log scale. So you're taking the log of the Boltzmann factor and you get back the energy. The energy is sort of linear in radius. And when you see that, it says, it doesn't have a cost for making the core, doesn't have much cost for making the surface, but it does have a cost for making a certain curvature, just from the fact that it's linear, whereas in the FOSS, all three components enter. And I can't go into the details of these fits. There aren't all that many data points, but we did do the analysis. And here's an example of extracting the parameters, but I don't want to go into details. And I just want to tell you two things about the implications of these two paradigms. One paradigm, clusters as fluctuations. They are energetically somewhat stabilized, but not all that much. And the other one, where the clusters are more energetically stabilized, and most of the proteins are in them. What implications might they have for liquid-liquid phase separation? And this is a speculation. It's a story right now. And you might have two ways to get liquid-liquid phase separation from these precursor clusters in the single-phase regime. Let's take this one. This is a typo. This is, I'm sorry, no. This is where you have the clusters. I described all that. Then you go the saturation concentration, where you get the micron size droplets, so above saturation concentration. But these clusters are fairly stable energetically, and very much possible that the LLPS has a substructure of clusters that have just aggregated. Now, just remind you of this plot of the timescales. These timescales don't vary very sharply at the phase transition. So that says that even above the coexistence temperature, in this case, in the LLPS region, you still have these three timescales. And that says, you still have these three conformations within the cluster. And so the LLPS large domains may indeed consist of clusters themselves. They've changed a little bit, we don't know, and this is something to measure. In the FOSS paradigm, where these clusters are fluctuations, it may well be that LLPS is a coalescence of these clusters, or proteins that were in their hydrophilic conformation. When you change the temperature or concentration, you go to the hydrophobic conformation. And this is the more standard view of LLPS, rather than coexisting substructures. However, these guys do have proteins in their amphiphilic conformation at the interface of the clusters. And it may well be that even the LLPS domains have those self-same proteins with amphiphilic conformations at the interface of the LLPS domains. And what's the implication of that? We will know that ampliphiles lower surface tensions. And let's get to that here, and this is my last slide before the things for. Okay, so these guys are decorated, we speculate, nobody has measured this, with ampliphiles, and this will also have the amphiphilic conformation at the interface. We then fit the data on the clusters, not on the LLPS, to get an estimate of this interfacial tension of the amphiphilic layer. And that interfacial tension is 4 or 5 orders of magnitude lower than the typical water-oil interfacial tension. So we have numbers, I think they're actually here. And the measured tension in many experiments is 1 micronewton per meter compared to water oil. This is millinewton per meter, much, much lower. Another paper measured both the tension of LLPS domains and also their curvature energy, their bending energies that are about 1 or 2 kT. That's also consistent with our fits. So we fit the properties of these clusters, not of the LLPS. We've gotten some numbers from our fits to experiment for the tension. The tension is indeed, because of this amphiphilic conformation, several orders of magnitude lower than a hydrophilic tension, and the bending energy is about 1 kT. So this amphiphilic conformation that has a slow rotational diffusion constant as measured may be at the origin of the very low interfacial tensions of LLPS domains. And the last thing I'm putting up here, but am I out of time? Yes. Okay, I'm out of time, but you can read it and we can talk about it during the questions. Well, this was a story. There were several anchors based on experiment, but a lot of it is still a story. And what can be done to make this story into scientific reality? One of them would be looking at the microstructure of the clusters, measuring their tensions, measuring their bending properties, seeing if there are more proteins that fit either of these two different paradigms. For people doing more molecular studies, can you see the coexisting conformations of one protein in solution that may have these 3 conformations? And this is exciting to me, and thank you for your attention.
Yeah, very interesting. I just wanted to point out, so we've done some measurements of these clusters, and so we looked at this with single molecule thread with Klaus Seidel, and we seem to see that within like 10 or 20 minutes, they do age, and they actually, you see alignment of beta sheets, so they're forming beta sheets within a very short amount of time.
This is in FUS?
With FUS, exactly.
The same conditions as Tony's?
Yeah, similar conditions, and so they aren't really stable, they're metastable, and then they form, so they seem to be off-pathway oligomers and not...
Right, right. Well, that indeed may be true. The CPEB-BB4, I think, are more stable on longer time scales because they're more stable energetically. I just want to point out something very speculative that's maybe interesting to you and related. If these proteins at the interfaces of these clusters or of the LLPS domains with the water have this amphiphilic conformation, that means that these proteins someplace have an oriented conformation. And that might be some harbinger of trying to make chains of these proteins or other ordered structures. So indeed, we're aware that these might not be the final equilibrium states, but it might be that having proteins that are oriented as opposed to proteins just dissolved in water or in the hydrophobic conformation may be important in how these proteins will then age. And that is an utter speculation, but we think that's maybe very interesting. Thank you.
So I just have a 2-part question, and one is, the first part is, like, it would be great to understand how this situation is qualitatively distinct from similar situations with amphiphiles and known to membrane science. And I guess maybe one very basic question is, how much larger are these proteins than a lipid molecule or your average amphiphile? And does this really impact quantities such as the bending rigidity of these interfaces?
Okay, they're both good questions. First question is, I think I know a little bit about membrane science from working for an oil company for 10 years. The difference, as I said maybe quickly, is that in membrane science, when you have such clusters stable, you usually have water in the oil, let's say oil in the core, and surfactant and water. Here, there's just protein and water and salt. So you have no oil, you have no surfactant. But the new part of this story is the same protein might act more hydrophilically, less hydrophilically, and amphiphilically. So that's It's very new. It's as if a soap molecule can then hide its hydrophobic part and be completely soluble. So that's what's new here. It's the same molecule doing that. The bending constant that we get here is extracted from fits of the light scattering data, and it's about the same order, and you're saying, but these guys are probably much bigger than a surfactant molecule. And a surfactant molecule, which is 2 nanometers or 3, well, they might not be so much bigger as you have to think of the radius of gyration, not the stretched length of the protein. So they're of the same order. But that is a good question. But that's what we got from fitting the data, that they have bending moduli of 1 to 2 kT, and that's what was also measured by the group of Law et al. In 2023, when they actually looked at the fluctuations of stress granules. It's a good question because the bending modulus goes like the cube of the length of the amphiphile. This is known from Landau-Lifshitz, and so it should be very strongly dependent. But these are not surfactants. They're more hydrophilic blobs and less hydrophilic blobs with, I think, much more subtle interactions with them than packing surfactants and interface. Those are good questions. The pressures? Oh, I know what you're talking about. I don't know. I think that there has to be work by people expert in protein conformation, single molecule, for trying to see these proteins. I don't think they will do the pressures, but I think that they will say what the conformations are. I know what you're talking about with the pressures, and I'm not sure that that's within their purview.
So I really like this, Sam. I think that
now you know why I asked you about
exactly. No, I'm so sorry. Right, you're like my cells. Yes, exactly. All the noses. So that, one of my takeaways here is that this is just what happens when you take one step from homopolymer, which is basically like only able to do kind of one thing, and suddenly it can occupy all these different conformations.
Right. We didn't model these as block. They can be models of block copolymers, and Gonain is doing that, but we're just looking at the mesoscale right now, and the EPR, which is the opposite of mesoscale.
Yeah. And so, of course, one of the questions I want to ask is the one that you don't want to answer, which is all about like kind of how long are the dwell times in these conformational states. So I'm not going to ask you about that, but if you want to answer, that's great.
That is not for theorists, I think, too. I think that's for experimentalists to measure the exchange times between the different conformational states. But it's known that in self-assembly, in membrane science, micelles, microemulsions, things like that, people do measure the exchange times of monomers in solution and the ones. So that's doable.
So instead, I wanted to ask you, you basically have shown with this simple system that you can get these 3 states, right? And you can then put together a different set of molecular conformations based on these 3 states. What's the repertoire that you can get out of these molecules, right? And the mapping from what the molecule can do to what the ultrastructure can do.
So I wrote someplace, and I emphasize, we have bypassed the conformational problem as theorists. And now we're aware of these 3 rotational states, but that's sort of like a symmetry argument for having those 3 things. That's all it is. But we have bypassed any theoretical detailed analysis because proteins are not homopolymers, they're not block copolymers, they're more complex. And that requires people like Dave Thurliminai to do more detailed experiments, but I had 2 references. People are starting to think along those lines. You're right, they might not only make an amphiphilic conformation with 2 blobs, you can get, what are called Gemini surfactants and surfactants which have 2 hydrophilic blobs and 1 hydrophobic. The sky's the limit. Or in some proteins, everything might average out. You may not get mesoscale hydrophobic and hydrophilic pieces, and then there'll just be proteins in solution, they won't make clusters. So maybe I should have said, thanks for your question, that there are 3 paradigms. One of the ones that make clusters rather weakly, one makes strongly, and others don't make clusters because they don't have the ability to separate their hydrophobic and hydrophilic amino acids into any appreciable distance. But for a theorist to answer that, it has to be somebody with an expertise at dealing at the amino acid level. But one of the great things about being old and being trained by people even older than me, and some of whom were very great, is that you learn to think about the mesoscale as what hints can it give you that might be a little independent of the more detailed questions. So this is just the beginning, I would say, as far as your question.
Yes, thanks a lot. It was a beautiful talk. I really enjoyed it. Maybe my question is related to the last one. I'm not completely sure. So you highlight the 3 different conformations in your scenario. Can you generalize it to many conformations, sort of even a continuum of different conformations between all these 3, and would it still work?
We did not do that. There are papers on phase separation. I remember in the old days one by Mike Cates, not on systems of proteins, but on systems of many types of particles. They separate when you get sharp phase separation, when you don't. We didn't do that. I'm trying to think what I want to show. I would say the 3 are tied to the anchor of the EPR experiments. And that's sort of, to me, a symmetry that there are 3 time scales. I think to look at the others, I mean, you can be a theorist and think about them, but maybe it would be good to have an experimental EPR study of other proteins and see if you get 1 time scale, 2, 3, 6, or 8. But it's an excellent question. We just haven't gone there. What we want to do is see, can we say something about these if we make a block copolymer model? It's a little bit of a trivial question theoretically because everything's known about block copolymers, but you have 1 block expanded and 1 more collapsed and things like that. And can we say something more? But I think the ball here is in the experimental court to see how much of the story is a story and to see if it's more general.
So one last question from Shira.
Thank you. Thank you, Sam. I think we discussed the question of different energy scales that have to be involved in this kind of picture. And I tried to count energy scales in this simple block copolymer picture, and the count does not seem to be satisfied. So I'm thinking that maybe to make your picture more stable, more plausible, maybe you should include 1 more degree of freedom, such as internal degree of freedom of amino acids, such as secondary structure. Maybe your blue and red are not only mostly hydrophobic and mostly hydrophilic, but there is some rearrangement of some switching between different secondary structures. Then it will be stabilized, I think.
Okay, so the first thing that Gonain is doing is saying, okay, I have a block copolymer, 1 hydrophobic, 1 hydrophilic. How do I map that to this picture? And that's what he's doing now. But your suggestion is very good. I'd like to hear more about your counting of degrees of freedom, because we thought that when we did the energetics of the cluster size distribution, no internal degrees of freedom, that these are the right terms that you would put in, bulk, surface, and bending, and the coefficients are related to your internal degrees of freedom.
Exactly.
These coefficients here are all of the order that for the whole cluster, each energy is of the order of kT. Per amino acid, if we take these typical energies, they're fractions of kT.
Okay.
So we did the fits. And when you do this thing times r cubed, so that whole energy is a kT. The next one is 2 kT. And if you divide by the number of amino acids, they're tiny.
Yeah, but you agree with me, Sam, that with energies of the order of kT or less, it's difficult to imagine that.
I know, and that's why we're not dealing with the amino acids, because the whole cluster energy is kT. But that we can deal with, like, from microemulsions and micelles. That's a very good point, but maybe it's consistent with your question about switching.
Thank you. So that's the end of the session, and now it's lunch.
Thank you.